Hey everyone. Thanks for coming out to our Biotech Talk. Um, Thanks for those of you who braved the Wisconsin weather, and thanks to those of you who are, averting your attention, diverting it at least towards this window for the purpose of this talk. So I appreciate that. I'm really excited to have Dr. Alex Veith here today, a postdoc in Chris Bradfield's lab. Comes from Baylor College of Medicine before this stop. Um, I'll try and keep it really short. Work a lot with Alex, especially as we've been describing some of the really interesting genome edited models we've made over the years. And one of the themes that seems to come up uh, quite a bit is this idea of using long read RNA sequencing to help describe these models. To better understand what these models are telling us. And it turns out that this feed is really not um, terribly trivial, and I think it's taken to sort of a special scientist like Alex, who has a really open mind, and he's a really good thinker to help kind of tackle this. And I'm happy that he's going to tell us a little bit about his experience in analyzing these data sets, and what it's told him. I'm not necessarily saying he you may not say he's a content expert in this thing, but he is a really good broad thinker. And so I'm happy to have him share with us the things he's learned along the way. So Alex, take it away. Thanks.
All right. Thank you, Dustin. Can you hear me? Okay. So I'm really excited to tell you about some of our projects using long read sequencing um, and how we're using it to identify unique transcript isoforms. So as Dustin said, sort of, I'm not necessarily an expert in sequencing or bioinformatics. I'm an end user of some of these technologies. And so I often rely on the technical expertise of the UW Biotechnology Center for their computational resources and sequencing expertise. I want to talk a little bit about splicing in physiology and disease, how we use long read sequencing for transcriptomics to identify these isoforms, and our use cases of using both Oxford Nanopore and PacBio sequencing. I can try to address questions during the talk, but I will also, any questions at the end would be fine too. So first, I just want to talk about this concept about alternative splicing is an important source of genetic diversity. Our genes are composed of different modules, or these exons that are spliced together to create our functional proteins. So splicing basics. Sorry, I just want to get my pointer. Specific sequences are recognized for their role in splicing. So when you have a splice junction, you have these canonical sequences that trigger the splicing proteins to recognize them, bind them, and create a splicing event. Now, for nomenclature reasons, we talk about a splice donor, it is the 5' end of an intron. So we're going to take our pretend gene right here with the exons are these colored boxes, and the line is just the intron that's going to be spliced out. We have sort of this canonical sequence again. This 5' end of the intron is the splice donor, which is where our splicing is going to the splice, that end is going to be donated to the acceptor, which is the 3' end of that intron. And so, we go from this red to this blue exon. Alternative splicing is a source of genetic diversity in eukaryotes. So for example, the first gene where this was really kind of described is fibronectin, which has a extra domain exon A, this EDA domain, that depending on which cell type this gene's expressed in, governs its solubility. So if it's expressed in the liver, it is plasma soluble and is involved in clotting. Another example of how splicing influences genetic diversity is the calcitonin-related polypeptide alpha gene, which shares 3 common exons, but then alternative splicing between exons 4 as well as 5 and 6 in different cell populations regulates the protein that is expressed. So for example, in the thyroid, we generate this mRNA and a pre-protein that is processed through proteolysis into calcitonin, while in neurons, a different mRNA is generated with, again, a different pre-protein that is proteolysed into CGRP1. Alternative splicing can also impact disease processes. So one disease that we are very interested in is neurofibromatosis type 1 or NF1 that can be caused by a spectrum of mutations. It's not a single mutation type of disease. But so I highlighted two examples where this group was, they were sequencing several NF1 patients. And so they found mutations that led to generation of these splice donors and splice acceptors. So for example, this mutation from A to G created a 5' splice sites that splice donor that resulted in a functional deletion in the mRNA. Alternatively, in the intron, they found a patient with this mutation that created a cryptic splice acceptor, which inserted nucleotides into the mRNA. Alternative splicing can also be used to subvert therapeutics in some instances. So for example, this is a cancer cell, HER2 positive cancer cell, where alternative exon splicing results in a different functional protein that is not recognized by targeting antibodies that can lead to tumor growth and resistance to different therapies. So long-read sequencing allows us to interrogate isoform level transcriptomics in a way that short-read sequencing could not before. So we're going to go back to our fake gene, where the blue exons are the canonical exons, the one that we anticipate to be spliced together, while these different colors represent alternatively spliced exons. In short-read sequencing, we can generate reads that cover across the entire transcript, but they're not linked. So if we're looking at this junction, for example, marked by this orange read kind of region, and this pink one, we can't tell whether or not a read from this transcript, is linked to this one. So for example, this orange read could correspond with either of these two transcripts, but this and different junctions downstream. Because these are linked in long read sequencing, we can definitively say whether or not a transcript contains different junctions and can provide a more complete description of that isoform. There's two long read sequencing approaches that we can use. The first is Oxford Nanopore. So I'm just going to show a bit. Not working. So, okay. Sorry. Video's not working. So Oxford Nanopore sequencing involves the DNA going through a pore that and how each nucleotide changes the ionic current through that pore that then signals a change in electrical signal that corresponds with different nucleotides. They can generate extremely long reads up to 100 kb. However, they are prone to error in homopolyramic regions. One advantage to this technology, RNA can be sequenced directly. PacBio is another long read technology where nucleic acids are circularized and they are deposited in these wells where when a base is added to the sequence, it emits a light that then is read and denotes the sequence. You can generate long reads between 10 and 25 kb. And the advantage to PacBio is that these hi-fi reads, which is reads where they circle over and over and over again, are extremely accurate. So how can we use this technology to examine splice isoforms? So I wanted to use some example data from our lab. So this is liver RNA from C57 Black 6 mice. We collected it as you would any other RNA experiment. So tissue was collected, stored in RNA later. It was isolated using Chiazol and ChioPrep mini preps columns. We diluted it to the specifications on the UWBC website. The sequencing gene expression cord did QC and library prep for our experiment. Sequencing cord used one ONT flow cell for 72 hours to sequence our data, and then bioinformatics core helped us do the trimming of the adapters and poly-A sequences. And the visualization, which I'll show you now, is on the Integrated Genomics Viewer, or IGV. I wanted to show sort of walk people through what an IGV view would look like. So I went through the literature and found a gene that was expressed in the liver that has alternative isoforms normally expressed. And so we found TPM1, which is tropomyosin alpha 1 chain gene. And so down here we see these are the ref-seq transcripts. So these are well annotated transcripts of this gene. The blue boxes indicate exons. So anywhere you see a blue box, that's an exon of that transcript. And the region between it is just the intron with an arrow pointing in the transcriptional direction. So to simplify this, this gene is going from left to right, transcribed left to right. Above, we have our actual reads for this gene. So when we look at an IGV view of our transcripts, and this is a splice-aware algorithm, so a read can map to multiple parts of the gene, and the coding, the regions that we have successfully mapped are shown in these gray boxes, with the introns sort of depicted as these thin blue lines in between the exons. So in our data, we see multiple transcriptional start sites indicated by these different exons, which again, we know from the literature. But we also see regions where we have differential splicing of these isoforms that correspond with the references as well. Another way to look at this type of data is through sashimi plots. So again, the bottom is the same ref-seq sequences I showed before with the sashimi plot above. And instead of looking at the sequences individually, we're essentially looking at them in aggregate, where these peaks represent coverage of an exon. So for example, we had a lot of reads that correspond with the 3' end of our gene. But again, as I showed the region where we can either have pretty much either of these exons, we see lower coverage. And these curved lines indicate the splicing pattern. So reads in which we splice from this exon to here, or from this exon to this exon. This can be useful to kind of visualize your data when you have a lot of reads that the other view, it's not very helpful. We can also use long read sequencing to identify transcripts with different 3' end processing. So I found this gene sort of just looking for random genes for another project, the CPN2 gene. And we see, again, this is our ref-seq transcript isoform. Because of the sequencing approach that we used, the 3' end of the gene is consistent. Sorry, the 3' end of the gene is well characterized. And so anytime we see this sharp pattern, that is where a polyadenylation site likely is. And so in this gene, we see multiple polyadenylation sites that I tried to go through the literature. This isn't really characterized at all. But we also identified this novel transcript isoform in which we start in the CPN2 gene, in exon 1, but then we skip the exon 2 of CPN2 and then begin transcription in this uncharacterized gene. I don't know anything about it. And so this is just another interesting use of how long read sequencing can identify novel transcripts in addition to novel information about your transcript of interest. So our first case study is a targeted sequencing project where we were trying to understand the transcriptional mechanism of an LSL allele that we generated. The goal of our project was to create a lock-stop-locks allele of the mouse AHR locus. So lock-stop-locks allows us to conditionally express a gene based on Krieger companies. And so this cassette consists of several components. These homology arms help direct that cassette to the genomic locus where it's going to homologously recombined. The LOX-P sites allow Cre-mediated recombination. The splice acceptor consists of an intronic region that then leads into a false exon, which contains translational stop codons. I just want to point out that right where we shift from intron to exon is the same canonical splice acceptor. And then we have a polyadenylation site. And the design of this project was such that we target to the first intron of AHR, the AHR locus, where then we would create a synthetic transcript and loss of function allele that could then be rescued with recombinase expression. We generated two founders. The first had the insertion exactly as expected. And we also generated a second founder in which we had a tandem insertion that was joined together, presumably by non-homologous end joining. These different lines had different phenotypes, and so that prompted us to examine how these LSL cassettes were functioning in both alleles. So I first used reverse transcriptase PCR to quantify downstream expression of the HR gene. I used sort of the default, the recommended primer probe set from TechMan, and which covers the third and fourth exon. I thought this would work well because it's downstream of our LSL and polyadenylation sequence. So we anticipated significant decreases in expression. However, we see at best modest changes that didn't really reflect the phenotype that we observed. So then I probed the first and second exon junction, which should be disrupted. And we see that the LSL cassette is incorporated and that junction is disrupted. And so we wanted to do long read RNA sequencing to try to understand the different isoforms that we were producing. So this is sort of a simplified IGV view that I want to show of the 5' end of the HR transcript. And so this is exon 1. And this is the tandem insertion, where I'm only focusing on the tandem allele for this experiment for right now, in which we have the left homology arm, the LSL cassette, so sort of everything in between those LOX-P sites, the right homology arm, and again, because this is the tandem experiment, we have the left homology arm again, the LSL, and the right homology arm. This line sort of corresponds roughly to where we anticipated transcription should end at the polyadenylation site, but we consistently see transcription continue past the polyadenylation site into the right homology arm, where it encountered a cryptic spliced donor. That then continues on to the second LSL cassette splice acceptor. And this helped us understand what was going on with our project and supported other data. And so we were able to conclude that the polyadenylation site used in our allele can be read through, again, explaining our downstream transcription, and it supports our other data. Our experiment did have some limitations. The prep we used has a 3' bias, and we think that because the AHR was relatively low expressed in these tissues, that's the reason why we didn't get we didn't get any full length reads, so we didn't have a lot of coverage of a lot of the 5' exons. So our second case study is sort of still in progress. It is single cell iso-seq of tumors in a swine model of neurofibromatosis type 1. I should have mentioned that iso-seq is, I believe it's a proprietary name for PacBio-based isoform sequencing, whereas Oxford, I believe, calls it just long-read transcriptomics. And so while in our last project we had a goal of specifically examining a certain gene, we went into this project a little bit more with an unbiased approach. So a little bit of background. Colleagues here in the Biotech Center and Dalton, they generated an NF1 swine model that was able to replicate some of the histopathological features of human NF1. These two tumors, as well as the normal adjacent tissue, was taken and used for single cell transcriptomics, where they were able to identify different cell clusters corresponding with fibroblasts, different immune cells, Schwann cells. And they found a significant number of the cells that they examined were fibroblasts, and that there were different subclusters of these fibroblasts. And these were the majority of the data in the experiment. So the idea in single cell isoform sequencing is that different cell populations differentially regulate isoforms of a gene. Now, and we kind of saw that with the CACLCA gene. And so that was sort of our hypothesis going into this, is that different clusters would have different unique isoforms. Others have also sort of hypothesized that tumors express alternative isoforms as well. And so we were trying to see if there were different isoforms between the tumors and the cells comprising those tumors, or was it just dependent on the cell population? There are several transcriptomic analysis pipelines. Several of them are, started by academic labs, while others are commercial, so PacBio and ONT. We're going to focus on the IsoSeq analysis pipeline from ONT. Sorry, PacBio, excuse me. The first concept to introduce with isoform sequencing is the idea of collapsing isoforms. So when you get your data and you have all your reads across the transcript, across the genome, you have to reduce them to the minimal number of unique transcripts. So how many unique isoforms do we actually see? And so this is called collapsing. So for example, these two reads share the same exon structures and combinations, so they would collapse into a single isoform. C and D, while different than A and B, still similar to each other, so they would collapse into another isoform. A and C do not share the same exon structure, so they would not collapse into unique isoforms. Or they would not collapse together. It's also true for A, again, just as an example, A versus a novel isoform E. When we're trying to think about how to do this collapsing, there are criteria we need to consider. First is this idea of extra 5' exons. This kind of comes up in single cell isoform sequencing because the kits used for the single cell transcriptomics either involve a 5' or mostly 3' ends, where that's where the cell barcode's attached. And so you inherently have a 3' bias where only reads that contain that barcode are included. And so we have to determine whether or not we should collapse reads that don't maybe reach the 5' end of that transcript. And so for single cell sequencing, typically we do this default collapsing where even though these two reads don't share the exact isoform pattern because they differ at a 5' end, they're collapsed together. We can also specify specific criteria within those differences. For example, how much of a 5' or 3' UTR difference are we willing to tolerate, as well as these fuzzy junctions. So if you get different reads that have single nucleotide variations at that exon junction, do you think they're likely to be a sequencing error or biologically different? And so most of the time, those are grouped together as single isoforms. Anything within, I think the default is 5 bases. Then sort of your first pass analysis is this structural categorization of your isoforms. They're relatively self-explanatory, but so we have our reference transcript, for example. A full splice match matches it perfectly. An incomplete splice match, sort of, as I mentioned, where it doesn't match it fully. Again, typically we see 5' difference. We have novel in catalog, which is a novel isoform with a combination of known splice sites. So in this example, this splice site is known. This is known. We're just skipping an exon. The novel not in catalog includes at least one novel splice site. So for example, while this splice site might be known, this splice site is not known in the reference. So that would be a novel not in catalog. There are reads that might just be intronic, so that's considered an intron, or reads that encompass genes. Reads that encompass exon and intronic sequence are called this genomic. These don't come up very often. We can also further subcategorize all these reads And so these are the full splice subcategories and incomplete splice categories. They're, alternative 3' processing, 5' processing, or both. And then the different types of fragments. The more interesting subcategories, in my opinion, are these novel in catalog and novel not in catalog subcategories. So they are a known combination of splice junctions, splice sites. So again, the difference here is this transcript, this teal transcript combines sort of junctions from both known references. Combination known splice sites, sorry. Intron retention, where we just retain part of an intron that should have been spliced out, or a novel splice site. So this was our starting point for the swine data. We looked at the classification of the samples as an aggregate, so just the normal adjacent versus the mass 1 and mass 2. And we did see a slight difference based on this novel in catalog classification. We were a little bit surprised to see that, our references were higher in mass 1 and 2. But so we wanted to look at these these were single cell experiments, so we wanted to look at them at a cell cluster level. So we broke our reads based on the cell barcodes that corresponded with the single cell experiment. We see that we have a strong concordance of being able to pull down the number of reads that corresponds to the number of cells in the experiment. So this top part of the figure corresponds with the number of reads that met each structural categorization criteria, while the bottom portion represents the percentage of the reads in that group. So for example, percentage of reads in fibroblast cluster 2 in normal adjacent. And so we see that one of the things that first became evident was the disparate number of cells and number of reads across cell populations might be contributing to our sort of sample level data. And so if you look at the percentages, we see that some of these have relatively consistent percentages across tumor or normal adjacent. And so we realized we had to group them together. So every fibroblast cluster 2, whether it was normal adjacent or mass 1, should be treated as one. This is sort of when we look at them in the subcategorization, we see that as well, that we couldn't look at tumor versus normal necessarily at the single cell level. Some of our interpretations we also realized were inaccurate because of our incomplete genome annotation. So for these classifications, sub-classifications, it's essential that you have a good reference transcriptome. So if you're working with things like human, mouse, Arabidopsis, you're probably going to have more luck using this. The swine annotation we found may be leading to a lot of these false, intron retention type of reads. So then we decided to focus more at the gene level. So we identified the top genes expressed in each cluster and then graphed their expression by the number of isoforms. So we're trying to understand genes that were low expressing, low diversity, or high expressing, high diversity. And so each dot represents a gene, and I've highlighted the top 10 expressed genes in each cluster. And so we then decided to focus just on the top genes in our entire data set to see how isoform number changes across cluster. So let's just focus on gelsolin, for example. We see that gelsolin is highly expressed in all of our fibroblast clusters, but the number of isoforms differ significantly across clusters. And so when we plot that data sort of as a percentage, this is where we started to realize that we need to treat this maybe more as a population type of analysis. So if you look at gelsolin across our fibroblast clusters, even though it may be comprised of 40 isoforms, 99% of them are one isoform in gelsolin, whereas we see more heterogeneous expression of another highly expressed gene, collagen 1A2. This has sort of prompted me to think about analyzing this data, as maybe as you would a microbiome data set in terms of alpha and beta diversity. So if you look at a gene like vimentin, we do see that across our fibroblast clusters, we do see, predominant one species, but that there may be a difference across the number of isoforms that are expressed at an alpha diversity level. We also started looking at our data in another way to think about exon representation. So one discussion we've often had is how important are isoforms that are detected rarely? And so this is a sashimi plot of collagen 1A1 and 1A2. And this is from that collapsed data file. So it is irrespective of the number of reads of each isoform is treated just as one. And so we see, here's our exons again, and then it's spliced across. This isn't a perfect comparison because we aren't incorporating the number of reads, but this was sort of how we first started to think about this data. And so this is how a new interpretation I'm trying to think about. So because we know that we have a 3' bias in our data, our most highly represented exon is most likely to be the terminal exon. And the lowest representation would most likely be that first exon. But we see in our these genes which are highly diverse, the exon level, it doesn't fit sort of a linear or even maybe a curve. So what we're now going to do is look at exon representation across our fibroblast clusters for some of these extracellular matrix proteins to see if that represents a difference that looking at our reads individually may be able to give us more information about the functional output of what we're seeing. So for example, we have any one read that skips these exons may not be relevant, but if you had 200 reads, 200 isoforms that occur once, that might be more informative. And so the next steps for this project are to focus on the ECM genes, looking at the diversity of isoforms and the exon representation. We're also going to look at Schwann cells because those are the most interesting cells in NF1, comparing them to a wild type reference. And our preliminary data says that alternative and processing could be an area of interest. So we've talked a little bit about genetic splicing, adding genetic complexity, how it contributes to normal physiology and disease states, how ONT impact bio can be used for long read transcriptomics, to understand specific gene isoforms. And again, this is sort of more my opinion, the transcriptomic level analysis of this data may need innovation because if you still look at data sets today, they still are mostly focusing on these structural categorizations that are not necessarily the most informative. And so how do we look at these different genes in a way that is a little bit more gene specific in an unbiased way? So I have to thank the Bradfield Lab, Rockefeller Lab, some of our collaborators, and a lot of people across the Biotech Center. So any questions? Thanks. Oh, so okay. This is a sashimi plot for tropomyosin right here. Probably. This is our mouse data, so it's just bulk. So I don't have single cell level resolution. But there are, for example, oh yeah, we could look. Um, yeah, that's the thing is our sequencing depth on this experiment was significantly better. Um, but it's still the same idea. Our data, again, this experiment has a three prime bias, so we see sort of this decrease. This gene is a little bit more complicated because it has several different exon ones, but these two exons in between are mutually exclusive. So if you express one, you don't express the other. So thinking about the exon representation across cell types or uh, states would is yeah what we want to do. Yes, the our ONT data had a significant three prime bias, and so we see a lot of three prime these trimmed three prime reads because this was a PCR cDNA experiment, so it involves reverse transcription with a poly DT tag, and so that's sort of our source of potential bias. Um, and, huh? Sorry. No, that's Pac Bio though, and we if we look at more highly expressed genes, we see a lot more full reads. It really depends on the gene you're looking at as to how well you're going to sequence across it. Because it also doesn't have anything to really do with size. You see, I mean, we see full length collagen reads that are 50 something exons. I want to do in our pig data set, I want to look at, I don't know how to describe this. So if we do our exon representation where our terminal exon is the maximum, and we do exon one as a ratio of the maximum, and we graph that as a ratio as a function of exon number of exons or length, probably both. Because you'd expect the longer the gene, the lower that first exon representation would get. Mhm. Nope. Only because that was the NF1 project. And so I think we don't, I'm not even sure we have RNA left. Um, okay. For O&T, it would have. For the AHR stuff, probably, but oh yeah. The pack bio for the swine, yeah. Yeah. You that's okay. I know what you're talking about. You're talking about this, right? Yeah, this was just a random gene we found. But this is ONT and not single cell. Mhm. Because we generate tons of data. But if you look at all of our genes, it doesn't matter which gene. It just ends up with this uh, tail. I would have to check that for you. Sure I can find that out. Oh yeah, I mean we got sizable genes that we got full length reads. And Mark looked at it. He said it looked fine. Um, what the? Okay. Oh, yes. So that is the next step. I was hung up on this full splice match, incomplete splice novel in catalog thing. And that's when we realized, with Harry Josie at the USDA, the pig annotation just didn't help us. So collagen 182 is our most highly expressed gene. And every single isoform is novel because it doesn't use exon 2 of the reference. So, okay, is that real or is that NF1 specific? So that's why, I mean, we also are going to take that. We decided we're going to take that wild type pig that was also sequenced and treat it as a bulk sample just to look and see what do we have reads that match the reference at all? And is that an NF1 pig specific effect? Um, and so that's sort of, yeah, that's our next step, my next step to try to do that. Thank you, Dalton. Great work. All right. Thank you.